Thank you so much for inviting me to speak here, and also thank you for running the title because it's quite a mouthful. I actually had to practice that a couple times to say it right. I will comment that I have not had enough time to practice this. We're going to see in a week, and we started a 90-bottle incubation study last night, so it might be a little bit rough. My apologies, but this is mostly work all for my dissertation, thinking across the California current system. The layout is focusing mostly on the Eastern tropical North Pacific and its intensification. We've got some background on its respiration and water masses there, some temporal variability on the P18 line, fault connections going northwards across the California current system, and then mesoscale variability, which is a little bit of a hint of our basin scale implications. We're going to take a jump to the other side of the California current system in Oregon. It's just a very small snippet, thinking about anoxia and iron supply. And then I'm going to sort of scale back and think about not my work, but other people's work, the basin scale implications of these findings. So, without further ado, here we see a figure of oxygen concentration across most of our planets, focusing mostly on the Pacific. You'll see there's this blue where there's high oxygen and red where there's low oxygen, about 200 meters. It really shows the oxygen deficient zones of the Pacific, and notice the California current overlapping with this northern one, which is going to be a heavy feature of my talk. We're switching the color bar here. I'm sorry, but this is an atlas of Pacific oxygen deficient zones. We're using 1 meter bin cruise data and Argo float data, basically looking at how frequently they observe oxygen deficient conditions. So blue means that very frequently, and it's basically permanently anoxic, versus red being permanently oxic, right? So this is basically how often do we see oxygen deficiency. This talk I'm focusing on the Eastern tropical North Pacific. It's the largest ODZ on our planet, and it's very important for the loss of fixed nitrogen, as well as habitat compression and ecosystem restructure due to the lack of oxygen and mobile predators, as well as greenhouse gas emission via nitrous oxide. And once again, it is the N member, or one of the main three, for the California current system down here. For metabolisms in ODZs, it's a bit different. On the left, we see a figure. We see a concentration on the top with 0 here, going up to the right. In blue, you see oxygen, and at the surface, oxygen is saturated the atmosphere. There's oxygen. As you go down in depth, there's this intermediate low oxygen waters, and then you asymptote again in deep waters. In yellow, we have nitrates, which is depleted in surface waters. It's being used biologically. And as you move down, you get this sort of asymptote in deep waters as well. In the open ocean, in this case, the main metabolism here is red-filled organic matter and oxygen are being consumed to release CO2 and organic nutrients. Now, even in cases where you can't measure the oxygen, this can still be occurring, right? This is very low oxygen, you can't even measure it, but we still have this metabolism being the primary one. An oxygen deficient zone is different because in these cases, you have nitrate depletion. So you have the loss of nitrates, right? And the accumulation of nitrates, which is the first intermediate in denitrification. And it's usually a very good indicator that we have denitrification occurring using high nitrate concentrations. And in this case, we still have organic matter, right? But microbes are breathing nitrates instead of breathing oxygen. And so there's a sort of metabolic switch that occurs upon low oxygen conditions, which can observe via nitrate depletion and nitrate accumulation. One of the first things I found in my PhD were the source waters for the ETMP ODZ. Here we have several cruises worth of data on the region and temperature salinity plots. You'll see that there's many points, but I want to focus on the ones that are darker, which are the low oxygen ones. Mostly here, the main water masses are the 13 degrees Celsius water, or the 13 CW, and the Northern Equatorial Pacific Intermediate water, or the NEPW, as I'll refer to the rest of this talk, because it's kind of a mouthful. Those are the important ones for this region of the ODZ. This slide is a bit complex, but it's very important, so I'm going to spend a couple of minutes on it. Okay. Once again, we're looking at that data from that atlas. So we have a series of 1 meter bin data, and this shows where the ODZ is. Now I'm looking at where basically the middle of it is, so the core of that layer. And we have this sort of triangle for the boundaries of the ODZ. And in purple is about the 26.0 isopycnal, so thinking 70 to 100 meters. And in yellow is more the 26.9, 27 isopycnal, so thinking 800 to 900, maybe 1,000 meters, depending on where you are here. And notice the ODZ is shallower in the northern region and the southern region, which we've known for about 70 years. But one of the findings that I had, and sort of how I'm going to set this talk up, is seeing how the source water paths into this region dictate the depth of anoxia, right? Where the actual oxidation layer is dictated by the source waters. We can see this using a ROM study of particles, basically backtracking, entering this ODZ region, which is done by Andrew Margolski. And see to the southern region here, that most of the entry is fairly shallow, around 26.4, with the hotspot here due to the Mexican coastal currents, around 26.2. This water enters the region oxic, right? It doesn't become anoxic until it's fully in the ODZ. So we have this oxic water coming in around 26.2, which means that the actual oxygen deficient layer is going to be deeper, right? Now, on the other hand, we have this NEPAW that's being injected by Sukiya jets around 10 to 16 degrees north, which is right around 10 to 16 degrees north here. And that's being injected deeper around 26.8 isopycnal space, right? So when this oxic water is injected into here, the ODZ, the oxygen deficient layer, has to be shallower, which is made of water from the 13C W, this 13 degrees Celsius water that's been moved all the way to anoxic up here. So the flow paths of these two water masses act as controls for the depth of the oxidation layer, as I'll talk about later. The strength of the oxygen deficient zone. Does that make sense? It would be a disservice if I went to a CLIFOR webinar and did not talk about one of my chapters, which was a 50-year analysis of data on the P18 line. So this is Aldebal's data sets going from 1972 to 2019 on the tip of Baja. Several of these are WOS cruises, a couple of them are CLIFOR cruises. There are 7 on this line. There's the 8th is a little bit off in 2016. It's marked as a Kuwait cruise. And this basically allows us to have a snapshot across this northern ODZ region. Across these years, of course, we are not well spaced, right? There's a lot of heavier spacing in more recent years than earlier, but still, it's a very impressive data set to be able to analyze. It's very fortunate to be able to do it. Here are those snapshots. So we're looking at a series of plots. On the left is 1994. We're moving over to 2019. Here. On each of them, we have the tip of Baja, about 22.5 degrees north, and we have 14 degrees north here. So north to south. And then we have potential density on the y-axis. We have shallow to deep. The color represents the amount of nitrogen lost. So we're thinking about basically nitrogen depletion relative to phosphates. We have an arbitrary 8 micromole per kilogram cutoff, which we use basically to visualize the spatial extent of it getting bigger or smaller. And the intensity of the color is the strength, so how much nitrogen has been lost. In 1994, I actually see the weakest ODZ since probably about 1600 AD. And across here from that 2012, it's shoals and also strengthens up here. So this intrusion into the 13 degrees Celsius water. After the shoaling stops, but it notably deepens into the NEPAW and intensifies. So these different mechanisms controlling the strength of this oxygen deficient zone. Right? Now we can take the top 10% of those values, basically how reprocessed the ODZ changes to the water that goes through it, and anticipate its strength or estimate its strength. And that dictates about 2.4 micromole per kilogram of fish nitrogen loss increase from 1994 here, 2012, and 2019. Also, the strength of this ODZ is really well correlated with the depth of 30 degrees Celsius water. This was previously suggested by a sediment/modeling study by Curtis Deutsch and Will Barrelson, but we had this water column data to follow it up, and now we actually have reasons for it. Notice that the mechanisms that cause ODZ strength are different for this sort of core ODZ, which is once again the northern region, versus the southern region with this very large intensification after 2016. The core ODZ strength, I have a good hypothesis for why it is. I have not looked at this, and I graduated, so I have not been looking at it recently. But here's the really fun part, is that the core ODZ strength here is very well anticorrelated with the mean option measured on CalCoffee data sets. So I just took the mean option from 100 meters to 400 meters for every single line, station, depth, etc., and lumped all together into an average for each season. average those and error bars there. So the error bars are seasonal. And see when you have a fairly weak ODZ, there's a lot of oxygen. Fairly strong ODZ, there's less oxygen. There's a sort of teleconnection between the anoxic metabolisms of Mexico and the oxic metabolisms off of California, which is interesting. Once again, the reason for that is because this specific equatorial end member water for the California current system is by the ETP ODC. These PWs are injected via the undercurrent jet and the countercurrents. They're transported into the California current via this California undercurrent, which runs along the continental margin and through the survey grid. And I'll highlight, here are typical plots of our three members. We have the Pacific subarctic upper water, which is sort of Gulf of Alaska water. We have this gyre water and PW. Notably, the water masses from PW are the same. So the ones off Mexico also come in off of California. Right, so that's the same water masses, the same impact. I did a different little data analysis of the Cal Coffee data sets, and I found that one water mass, that deeper one, the NEPW, off Mexico is responsible for 80% of the loss of oxygen, which is about 1 micromole per year across that data set. So that's a very important controller on that. But if we take a step back here, the real conclusion to this talk that the ETP ODC strength has increased about 1.3 times between those years. Those drivers were significant, but I really want to focus on the 30 degrees Celsius water and its depth. Because in the ETP, having a shallower 13C water increased the organic matter supply into the ionic waters, but in the California current system, it also changes the ratio of water masses in the undercurrent. So when you have a shallower 13C water, you have more NEPIW in the undercurrent, which has lower oxygen in that region, and supplies less oxygen. So that's those connections. We have this basically master variable of this water mass depth across this entire sort of semi-coastal region, or marginal sea region, right? One of the big takeaways from that time series of 50 years is that we cannot yet claim that climate change has caused the intensification in the core ODZ. I compare that against sediment data. I don't have time, unfortunately, to talk about it in this talk, but if you're interested, I really recommend you look at it. It's really cool. I'm very proud of that analysis. So next, I'm going to take a step back and hint at why these sort of variables are important for a more basin scale climate approach, which is analyzing data from the Roger Revelle. It left from the tip of Manzanillo here and went west through the ODZ. On this transect, we observed nitrate accumulation. Notably, it's not like a blob, right? It's sort of string of pearl distributions. There's nitrate hotspots. And if you look at the oxygen on the same transect, this is from 0 to 25 micromole. There's not really a difference between like here and here, right? Or here and here. Like you can't really tell an oxygen space why there's nitrate there and not there. So turning to water masses, here we're looking at the contents or the relative ratio of 13C water to other water masses. And notice that in each spot with nitrate accumulation, there's also a hotspot in 13C water for every single one of these. So the 13C water leads to this sort of accumulation in nitrate. Why is this important? And is that there's sort of these blobs that are moving here, but it doesn't really answer my question for why do we have this string of pearl distribution in general. For that, I turn to sea surface height anomaly. So for that, we're looking at basically subsurface mesoscale features can cause ripples on the sea surface, which you can observe via satellites. This is a GIF I took with chrome capture of Copernicus, and I drew the cruise track on just via computer mouse by hand. Well, notice that each for these hotspots of 13C water and nitrites fall on the sort of mesoscale features that are moving off the continental margin and into the basin. So in this, we have these eddies that are enriched in a specific water mass that runs on the continental margin and contains sort of sediment drag material. And they also all act as hotspots for active cycling that are moving off of the continental margin. So in each case, we see the nitrites in them, suggesting there's stronger denitrification in each, right? And those are all being funneled off into the basin. I'm going to take a quick hop, skip, and a jump to Oregon. The real motivation here is deoxygenation and how climate change causes that. So if you look at historical oxygen data, or its depth versus oxygen concentration, and there's a map of those points, the big takeaway is in 2006, we saw the emergence of anoxia in the current system for the first time that we've reported. And this change in chemistry from oxide to sulfidic caused very significant fishery die-offs. Right? So that's one of the big modifications for the study. But also, low oxygen has this really interesting impact of changing constant margins from iron sinks, where iron precipitates out as oxyhydroxides, into iron sources, where if you have low oxygen in the sediments, you have iron reduction, and low oxygen above in the bottom waters, that iron too can persist and be transported before it gets reoxidized. So for this, we're going to move over to this northern part of the California current system, specifically looking at these 3 stations. And lo and behold, if you go during summer hypoxia, you see hypoxia on the seeded bank. Here we have profiles, depth in these black lines where we sampled. We have oxygen where white is hypoxic, about 20 micromole each place. And we see very, very high iron. This is dissolved iron measured by my lab mates. The color bar does not do this justice. It's actually about 30 nanomolar, which is very high for the ocean in both these places. But we do still see about a 5 nanomolar export of iron on the constant margin. Why this is important on a large scale is that we also took out an ADCP on this ship, and we could see this transport of material on the shelf break. So we have zonal velocity on top and radial velocity on the bottom, where this blue region on top shows a strong off-shelf transport, which is likely linked to the very strong California undercurrent we saw going north of the same region. So these column margins are enriching this water full of iron that is fairly stable and persists. The conclusions here is that we have this sort of self-hypoxia, which transforms the column margin into an iron source versus a sink, and we have these undercurrent dynamics that can move this iron off the shelf. So now I'm going to present stuff that is not my data, but is why we should care about this sort of lateral advection and mesoscale processes for a larger Pacific storm, which is thinking about these forward undercurrent eddies. In all cases, we have this constant margin with low oxygen waters in the coast, but when the undercurrent runs along topographical features like sort of bumps or banks, it can spit off these sort of mesoscale eddies, right, which have water that reflects the constant margin, which is more organic matter, a different redox system like nitrogen producing, iron presence, etc. And analysis of the global RO database is that we see a lot of these undercurrent eddies, which are in red here, spinning off counter margin and sort of focusing towards Hawaii, which is sort of interesting, but crossing this specific basin. So we have these waters that I've been talking about, right, with this different processes going all the way out here. Christina has also looked at this via modeling and found that you can see the precession of ocean anomalies moving southward in latitude over time at different depths, right? So these eddies also explain ocean variability in the Pacific as well. So with that, I think I'm pretty close to time. So the conclusion is that we have these sort of local or longitudinal forcings with regards to the 13 degrees Celsius water depth and its impact across this basin, which then change the biogeochemistry of the near shelf or shelf waters with regards to iron supply or nitrogen cycling. And then those are exported across this basin and have a very significant impact on the oxygen budget, as well as the microbial dynamics and nutrient dynamics in the Pacific Basin. With that, I'd like to acknowledge the funding that I received during my PhD and advising during my PhD, as well as some of my fellow lab mates at time periods. Thank you.
Hi, yeah, I guess I'll start. Thank you. That was a really interesting talk. I was curious about, so when you were showing the eddies off of Barra that had a signal in sea level, are those, they must be connected to the surface, right? So is it just the advection of water from the coast, or could there be processes at the surface, like changes in production, that would also impact what you see in nitrate and nitrite at depths?
So I would say that in these cases, there's a very strong salinity feature. I'm going to go way back for this one. But there's really, the reason why there's oxygen deficient there is that 13 seawater sets the oxycline depth, right? So there's a very switch into the bottom thermocline set by this water mass. So I would argue that for these eddies, that is material that is laterally transported rather than vertically sinking from the surface. And I don't think I have the slide in this deck because I sort of threw this together, but you can actually look at the isopycnals as well, and you do see bulging of those, right? So it's sort of contained material moving off shelf. To get to the other question about supply material, the other interesting thing is that if the eddy is strong enough, so we went through one really strong one on a different cruise, and you actually can see oxygen being pulled in at the core. So you have really strong nitrate signals, and you have to turn on the other side of it, but you see oxygen getting pulled in, and the loss of nitrate just in the core of that eddy. So that's, I'm sort of arguing that only in the really strong cases do you see that material from vertically transporting in that little hotspot. Does that answer your question?
Yeah, totally. Thank you.
Yeah, thanks very much. That was a great introduction. Yeah, thanks for inviting me to talk. So I'm going to try and focus on a couple of different aspects of the ocean carbon sink and the biological pump, and how we can link observations and models. Observations, obviously, to try and help inform parameterizations and calibrate what we're seeing out of our simulations. How do I shift that on? There we go. Okay, so understanding the ocean carbon sink is obviously really important for society as well as the environment. So there's been a bunch of different attempts to try and recreate the ocean carbon sink using observational products. And if you plot a time series of those, they kind of sit up here somewhere. We have used ocean biogeochemical models for sort of 30 or 40 years, running them forwards to try and get a handle on the ocean carbon sink. And if you plot those next to each other, they kind of come out down here somewhere. And so the difference between what the models would suggest the ocean carbon sink and the observations suggest amounts to about 1/3 of the total overall amount of uptake of carbon in the ocean, which is hugely significant. And like this tweet that I used to motivate this talk, we've got a lot of work to do, and it's causes some nightmares in some aspects of our work. But the ocean carbon sink is really a combination of different effects of the physics, the biology, and the biogeochemistry of the ocean carbon cycle. And what we need to do is to be able to mechanistically unravel those different dynamics and use observation and models to quantify the different fundamental and/or missing processes that are in our models, and to try and close that gap. So today I'm going to talk about a couple of things. The first is Eco-Darwin, which is an ocean biogeochemistry and ecosystem state estimate, and to look at some interesting results from that. This is using, oh, it's not playing, there we go. So it's using observations to calibrate the model machinery based on, you know, the model that we have, and reconstruct the ocean carbon sink. The second is like going a bit beyond the models that we have and looking at idealized models of trace metal cycling and the climate impacts of those. And this is to really get at that aspect that, you know, our parameterizations are imperfect, and some of them may indeed not include important processes and feedbacks that may have significant climate effects down the road. So I'm going to talk about some of that. And then finally, uncertainty overall in the biological pump. If you kind of, ocean carbon cycle in a nutshell is basically what goes down usually comes back up again. So primary production in the ocean surface, which is limited by sort of nutrients and iron and trace metals in different regions, form sinking particles. Those sinking particles drift down and are remineralized by bacteria and reprocessed by higher trophic levels, which gives you something of a profile like this. And eventually, I say usually comes back up, but eventually at some great depth, there is a small proportion of carbon that actually exits the system into the sediments and gets sequestered for really long timescales. And it's that kind of carbon that people are aiming for to try and lock away anthropogenic CO2. The first aspect is Eco-Darwin. And So I'm showing a animation of air-sea CO2 fluxes at probably quite high temporal resolution. You can see all the effects of the winds and stuff like that that the model is forced by. So Echo Darwin is a combination of the Echo estimating the circulation and climate of the ocean project, where you have they have a physical model that they integrate as many observations, particularly since the age of satellite altimetry, using an adjoint-based methodology, which allows them to adjust various parameters and initial conditions and forcing to get a really good estimate of the state of the ocean. On top of that, we overlay the Darwin model, which is an ecosystem model developed at MIT, which has the full Darwin model is kind of very complex and very expensive. So we have a kind of intermediate complexity version of Darwin, which has 5 plankton functional types, 2 zooplankton types, and they all interact with each other and with the ocean carbon cycle that we solve explicitly. The biogeochemistry side of this is optimized not using the adjoint, but using a Green's function approach, which allows us to optimize what's quite a complicated bit of machinery in the Darwin model. We compare, sorry, cut out there a minute. Yeah, so we optimize a variety of different parameters, including stuff to do with the ion cycle, phytoplankton growth rates, palatability, and initial conditions. And we compare the model state to SOCAT, Ocean Surface PCO2 Atlas, profiling bioargo floats, which give us nutrients and oxygen. And then we can derive DIC as well using the carbon system solvers, and as well as the GLODAP profiles from ships, which again give us a bunch of nutrients and carbon and alkalinity. So this is really a project that's spearheaded by Dimitri Minimelis at JPL and Dustin Carroll, as well as the rest of the ECHO team. So thanks to them, and thanks to also everyone who's contributed data to these different archives, which without that would be impossible to do this exercise. So the key result from Echo Darwin, I guess, is that the ocean carbon sink that we can calculate from our constrained state estimate is entirely consistent with observational-based estimates of the ocean carbon sink itself, which is great. And bearing in mind that that is within the machinery of the model that we have. What's also nice about using the adjoint approach for the circulation and the Green's function approach for the optimization of the biogeochemistry is it gives us closed budgets for all the different tracers, so heat and salt and carbon, which allows us to do a bunch of interesting things to separate out the different drivers of the ocean carbon sink and air-sea CO2 fluxes. And we can look at that across time and space as well, because the model simulates that everywhere. So what I wanted to point out here was some interesting ENSO variability that we've come across. You can particularly see the sort of 1997 and the 2015-16 El Niños in the Pacific. And then we can tease apart what is causing these different tendencies in DIC. So for example, one big thing that you notice is that the air-sea CO2 flux actually stops outgassing and almost becomes a small uptake. And what's basically happening is that is associated with the changes in upwelling of carbon from the deep ocean. That upwelling brings nutrients with it, which drives this biological uptake, and you can see that that kind of falls off at these different El Nino events. You can then split the global average or the Pacific average that I showed into the Western Equatorial and the Eastern Equatorial Pacific, and then you get these opposing effects, which are really interesting. So in the Eastern Equatorial, it's pretty much the same as the average, which is the you get a decrease in the air-sea exchange, a decrease in upwelling of DIC, and you can kind of see that in the 97 and the 2016 events. And then you can kind of also track the change in biology. You can see that there's a sort of decrease in biological activity at the surface, which also manifests further down in the water column. as the particles kind of sink out and are remineralized. But the reverse actually occurs in the West Pacific. So you can see that the biological activity is actually increased, and it results in a slight strengthening of the biological pump there. So that's kind of cool. Having done a global sort of ocean breakdown of the different drives of carbon, we can now also link that to what happens at the very sea surface. So we can get this full kind of integrated perspective on where the carbon is going and how it's getting there. So this is a method that I developed where we can partition the drivers of the air-sea CO2 fluxes into, so this is the net CO2 fluxes. There's a part that is due to air-sea heat fluxes, where warming at the Central Pacific increases the temperature, which decreases the carbon saturation and drives an outgassing. Conversely, where we have lots of biological activity, that drives an uptake in CO2. And particularly in the Southern Ocean, you can see that where you get upwelling of carbon and nutrient-rich deep waters, those nutrients drive primary production and carbon outgassing. Primary production in the Southern Ocean is kind of limited by light and iron in particular. And so not all of that carbon and nutrient that is upwelled to the surface gets utilized biologically. And some of that carbon is allowed to outgas to the atmosphere. And that's basically what the disequilibrium part is showing, is that there's a mismatch between the biological uptake and what's being upwelled to the surface. So that was a kind of quick, oh, okay. So, like I said, the state estimate that we get from the Echo-Darwin model is kind of dependent on the machinery and the code that we have. And so obviously we'll want to be able to improve that and get an even better estimate of the ocean carbon sink. So one of the ways that we're going to do this is to assimilate ocean color observations and other data sets such as particulate export fluxes, geotraces, metals, for example, and other such information, which will allow us to tune our model better. But also we can Darwin model actually has a radiative transfer model as part of the sort of machinery itself. And so we can generate hyperspectral information, which now we may be able to use new observations from NASA's PACE satellite to try and link those things a lot tighter and get a better mechanistic understanding, particularly even plankton functional types that we can observe from the satellite, as well as look at where they are in the model and make sure that we're getting, you know, who's living where and for the right reasons. The other thing I just wanted to mention is that we're also looking at integrating a bunch of new observations. So there's this new project that I've started working on called Explanations, which is a UK USF collaboration, where we're going to integrate observations from the rapid and OSNAP constraints into the physical model themselves, and then look at how those how that affects the carbon cycle, the carbon sink in the North Atlantic. And you can kind of see our sort of an initial analysis of a slightly different version of Echo than what I showed before, Echo version 4. Compares interestingly to the OSNAP array in terms of overturning circulation. So you can kind of see that it gets some of the variability right, but not necessarily the magnitude of the overturning. So we'll be able to hopefully integrate some of these new constraints into the model to improve the North Atlantic carbon sink there. And then, of course, we can do a full mechanistic breakdown of the carbon sink, and we're going to try and have a really good quantification of the uncertainties that are associated with that, and with these new, bringing these new observations on board. So now I want to move on to a new kind of parameterizations and stuff like that I think maybe would be an important aspect to include for slightly longer integrations of the model. And particularly, I don't think that any model, any model or story about the ocean carbon sink is complete without mentioning trace metals. So in a bunch, just a quick overview, that in a bunch of different regions, you get elevated macronutrient concentrations, which if you go to these regions and you amend those waters by adding different trace metals, particularly iron, which is shown in the red dots, you can stimulate production. And vice versa, if you go to these dots in the sort of subtropical gyres and you add additional macronutrients, that generates growth. So those regions there are macronutrient limited. And so iron limits production over about a half of the sort of surface ocean. And so you could think, okay, well, what if we go to those places and we dump a bunch of iron, that could generate a bunch of CO2 drawdown? And you can do that in a model. So here's one kind of experiment where Steph Dukovic did that. And you can see that, yes, indeed, you go to those places, you add a bunch of iron, you can generate CO2 drawdown, but it's not necessarily a one-to-one link. So you can see that the Southern Ocean, although it has vastly limited, vast regions limited by iron, actually you don't get a huge amount of carbon uptake compared to, say, the equatorial Pacific, largely because other things become limited. But the iron cycle is still really incompletely observed, and there's still a lot of room for expansion on what we know. What we do know is that iron is really insoluble in oxygenated seawater. And so over 99% of the iron that you can measure and that is available is actually complexed or bound with, associated with organic molecules called ligands. And those ligands are produced by biological activity. They're all organic. They're either produced actively, either as siderophores, which are produced by bacteria to competitively trap iron, or phytoplankton cells exude a bunch of polysaccharides and other stuff that kind of floats around in the environment. And that can also interact with iron and prevent it being lost by scavenging. Further down in the water column, you can get accidental production, what I call accidental production of ligands through cell lysis, and that kind of releases the internal cell contents that could bind iron in life and now is allowed to kind of float around in the water column. And a small, tiny portion of that could act as ligands to protect iron. And so the nature and sources of these ligands are really important, and cycling are really critical questions that are being kind of actively researched. And so you can, there's kind of two ways to approach this. Either you have a very simple kind of scheme to parameterize ligands in that you have a constant concentration everywhere, or you can have a really dynamic, complicated biological linkage with tens of parameters and all sorts of different stuff going on. But the approach we wanted to take was to kind of think, okay, how can we calibrate a parameterization of ligands using the observations from GeoTraces project? And so we came up with this kind of 2-parameter scheme. We've got a fraction of organic matter that interacts with free iron as a ligand. So that's the production, and then a degradation timescale. Because ligands are generally labile, tasty organic molecules that bacteria want to consume. And if we implement that scheme in a kind of idealized box model, we can see that the ratio of those 2 parameters depends, sort of controls the regime that you might, the ocean regime that you find yourself in. So if you have high ligand, low ligand production and high ligand turnover, basically you don't get any ligands in solution, which means that your iron is scavenged and lost really quickly. And so the entire ocean is macronutrient replete and iron limited. On the other hand, loads of ligand production and very low timescales of degradation mean that ligand builds up in the system and you trap all the available iron, or a large percentage of the available iron, which means that the entire ocean becomes macronutrient limited instead. There's kind of a region in the middle, and when you compare these box model simulations to those observations, sort of averaged in a certain way, what you find is that the model skill is high in that co-limited regime. So in some regions, you get ligand production, and that stores iron, for example, in the North Atlantic, and then the ocean circulation redistributes that complex iron into the Southern Ocean, and the iron is upwelled in the Southern Ocean where there's lots of macronutrients and kind of generates some production there. Anyway, so the skill of the model is also much better than a model with this fixed ligand scheme. So that kind of generates a good fit with observations that we have for the modern. But what about in the future, where the MOC, the overturning circulation, is projected to decline, associated with, particularly in the Southern Ocean, associated with glacial runoff from Antarctica due to melt, and also in the past when the MOC is thought to have been reconfigured, such as in the last glacial maximum. And so what I did was I did two sets of ensembles where I had fixed ligand concentration and ran with a different strength of Southern Ocean overturning circulation. And that gives us this curve. So as you strengthen Southern Ocean upwelling, you increase the amount of carbon and nutrients delivered to the surface. Again, production is limited in the Southern Ocean by iron, so a bunch of that carbon outgasses. And the stronger you upwell, the more carbon outgasses to the atmosphere. You get this positive relation. Sorry, jumped out there again. So yeah, we kind of known about that for about the last sort of 40 odd years. But now if you turn on the variable ligand parameterization, what you find is actually the opposite relationship between the MOC strength and atmospheric CO2. And so basically, when you strengthen the circulation, you're bringing more carbon and nutrients up to the surface. The more nutrients actually stimulates production a little bit, which then generates more biomass, which generates more ligands and can trap more iron. And more iron allows more biomass to reproduce from the excess macronutrients, which generates more ligands and more iron, and so on and so forth. So actually, when you increase the Southern Ocean upwelling, you now result in a very strong drawdown of atmospheric CO2. And the reverse occurs when obviously you decrease the supply of nutrients, which decreases the amount of biomass produced, decreases the quantity of ligands and the availability of ligands, which reduces the amount of iron that's protected from scavenging, which carries on this kind of feedback loop. And so in this state, there's sort of global iron limitation because ligand concentrations are very small, and you get generalized outgassing, which is kind of really dynamically changes the relationship between ocean circulation and atmospheric CO2. And you know, I'm not sure that many models include such a feedback. And so for the modern, we're probably okay, but then going forward, you know, whether we see an increase in the strength, an increase in the strength of the natural carbon sink when you decrease the MOC, or an outgassing, a decrease in the strength of the natural carbon sink is an open question in my opinion. I'm going to skip this, but just to say that we're putting this feedback into a global model, and we actually see very similar behavior, at least in terms of the modern state and comparison to the geotraces observations. So the third part I wanted to quickly go through was looking at the uncertainty of the biological pump. And so most models, a lot of biogeochemical models either include an explicit Martin curve type parameterization, where you decrease the sinking particles by this power law with a B value that you kind of determine, or you kind of have a sort of implicit Martin curve where the dynamics associated with the B and the power law, such as increasing sinking velocity with depth, or decreasing remineralization timescale with depth, which would both give you a power law. Yeah, and so what we were doing in this model was to look at how much of the uncertainty in the biological pump is associated with the uncertainty in that value of B. And so there was a couple of studies where you kind of they've looked at particle distributions throughout the ocean, and come up with a range of B values of between 0.7 and 0.98 as kind of a sort of one standard deviation each side of a mean. So, and then also there are lots of other ways that remineralization might mechanistically be occurring. And so we tried to, my co-author, Kale, at the National Oceanography Centre, with Kelsey Bisson, had written a paper where they kind of tried to distinguish between different mechanisms of the biological pump using observations, but found that they couldn't really tell which one was, you know, the real thing. And so there's a bunch of different mathematical forms that those can take: exponential profiles, double exponential profiles, for example, one that includes the effect of ballast, so calcium carbonate. And we tried those, we fit these different mathematical formulations to the control Martin curve. And so this is what, in 3 different ways, so you get a bunch of different curves. But basically, the important thing to take home from this is that some mathematical forms allow you to get depth-dependent attenuation of particles, such as the power law and the variety of different the ballast and the rational form and stuff like that. But the exponential remineralization profile has depth invariant attenuation of particles, and the gamma profile, which is from a paper by Dan Rothman, has kind of not a huge amount of variation in depth dependence of attenuation, particularly for this experiment here where we fix the folding depth. Anyway, when you run that in a global biogeochemistry model, what you find is that the parametric uncertainty of the biological pump is about plus or minus 25 parts per million in terms of atmospheric CO2. So varying B by between 0.7 and 0.98 generates that amount of CO2. And most of the other profiles that we tried actually fit within that uncertainty. So that's quite good. If you take an average of those, you get to something like the structural uncertainty associated with what mathematical form you use for the biological pump is about 10 or 15 parts per million. So it's roughly a third of the overall uncertainty of the biological pump. And then what's really important is that you get this depth dependence of attenuation, because if you don't have that, then you generate really large atmospheric CO2 uncertainties well outside the range of the parametric uncertainty of the Martin curve. And that's kind of important, because if you're not remineralizing, you're sinking particles in the right depth levels or into the right water masses in density space, that really affects the sequestration timescale of the CO2 that you're putting there. So being able to figure out A, how to get the carbon down there, and B, where it should be remineralizing, really affects how long the CO2 is stored there. And then so Helen mentioned that I'm working on this carbon cycle model of the future, I like to call it, with a postdoc Hung-Dee Lang, Emily Zakem, and Raf Ferrari and Greg Ragnar. We're basically rebuilding from scratch a biogeochemical model in Julia, the Julia language, which is optimized basically for use on GPU hardware. And the GPU is incredibly fast and efficient. So we can now, the physical model, Oceananigans, actually can generate a simulation of 10 years per day at 10-kilometer resolution using only a very small amount of resources that are available on the sort of largest HPC systems, which means that we can now run really high mesoscale resolving simulations in zombles of those, which is absolutely amazing. So the goal is to have this new BGC model embedded in this really efficient, fast Oceananigans model, and be able to, part of the CLIMA goal is to assimilate in near real time, all available observations, and for us that would include both, pace and BGC Argo, different ship profiles and stuff like that. And also by building it from the ground up, we can reassess a bunch of different decisions that have been made over the last 40 years to build BGC models, and try to reduce the uncertainty in the ocean carbon sink even further. So I'm just going to leave that up there. Basically, trying to build a bunch of models and make sure that they're constrained and informed. The parameterizations in those models are informed by observations in order to get a handle on the ocean carbon sink. So thanks for listening. I'm sorry my internet was a bit dodgy. I'm happy to take any questions or bits that I might have skipped over.
Yeah, hi. This is a great talk, and actually the second talk I went to this week about the importance of really understanding the ocean carbon sink. I'm wondering, I think based on the work that you're doing and the summary that you're doing, both looking at the trace metal feedbacks and developing this new carbon cycle model, if you think that you would be in a position to think as well about potential effects of different types of MCDR, and whether or not you think that that's something that new carbon modeling will be better able to tell us what some of the impacts of that might be.
That's a great question. I think MCDR is obviously becoming increasingly popular to try and apply biogeochemical models to, and I think that I don't know. Yeah, I think that trying to apply these new models, they're still going to have possible deficiencies in their setup based on, we can't necessarily include every like method that people might want to deploy. For example, I'm not sure how well the models would go from simulating phytoplankton and zooplankton, and then to try and apply them to, say, a macro algae kind of approach. So I'm not sure exactly how well these models would work for that, but I think these models would be perfectly suitable for the more MRV side, so the verification of the amount of CO2 that has been drawn down. I think particularly with the data assimilations aspect of things, we should be able to at least provide a good counterfactual simulation in which to try and, I don't know, determine some of the CO2 drawdown. I think yeah, that's possibly a bit of a long-winded to say. Yeah, sure, possibly.
Yeah, thanks very much. It was a good answer.